Again, I'm truly feeling very excited to have the opportunity to share with you what we're doing here at Vizgen. And today's topic will be really focusing on explore new dimensions with spatial genomics. I hope to use this opportunity to really give an overview of our platform technology, MERSCOPE, how it differs from some of the other spatial genomics technologies. And at the end, I will also use a couple of showcase examples to really illustrate how you could potentially use this very powerful technology to discover new biology. So first, to really give some background and context first of what I'm talking about. So let me just minimize this window here first. So as many of you know, biology questions or biological tissues can be very complex and are made of many different cell types that will express genes very differently. And over the past 2 or 3 decades, sequencing has really revolutionized our way of understanding different biological systems. As you can see from the landscape of genomic evolution here, from bulk RNA sequencing to single cell sequencing. Nowadays, we really have the capability to interrogate the gene expression at single cell resolution now. However, as you can imagine, sequencing-based technologies actually have some fundamental limitations because they don't really provide any sort of special context of what you're looking at. Because in order to do sequencing, you either have to dissociate the cells or destroy the cells, and by doing so, you will actually lose the very rich spatial information. And because of that, you can imagine how important actually it will be to have a tool that will enable this spatially resolved single cell measurement with a very high multiplexed measurement capability. And that's actually what Vizgen is working for. Specifically, we are commercializing a technology called MERFISH that really enables in situ transcriptome imaging. And our commercial platform, MERSCOPE, pictured here is the complete solution for MERFISH technology. So in the next few slides, I feel like it might be very useful for me to sort of really explain the technical background of what MERFISH is and why, how it differs from some other technologies. And as some of you may know, MERFISH was originally developed by our scientific co-founder, Dr. Xiaowei Zhuang at Harvard University. And it stands for multiplexed area-robust fluorescence in situ hybridization. So this is an RNA imaging-based technology that will really be able to allow researchers to locate and quantify hundreds, even to tens of thousands RNA species at the same time with subcellular resolution. And it is built based upon single molecule fish, which is pictured on this slide. So as shown here, many of you may have some familiarity of single molecule fish. So in order to perform single molecule fish, different RNA transcripts are first labeled with these fluorescently tagged oligos. And by directly counting the transcripts that shows up fluorescence, then it allows us to really directly understand and quantify the exact copy numbers of those RNA transcripts. So the limitation of single molecule fish is that the gene throughput or the plex number is very limited, often less than 5. This is because it relies on the spectrally distinct dyes to report different biomarkers or targets. And since sometimes we don't have hundreds of different spectrally distinct fluorophores, then it is difficult for us to sort of really do 100 or 200 color imaging for different RNA species or biomarkers. And to really overcome this fundamental limitation, MERFISH does it a very different way. Basically, it implements a new complementary barcoding scheme that now enables for simultaneous image for hundreds to even thousands of RNA copies. So I'm going to use the next few animation slides to explain the principles of MERFISH. But briefly, we label the different RNA species with a tile of oligos, and each of them contains a binary barcode. And then through multiple rounds of sequential imaging, as illustrated here, by flowing in these readout probes, then some probes in certain rounds will be highlighted and turned as fluorescent on signal. For example, here, this particular RNA transcripts in the second round of imaging, it will be fluorescent on, and then it will give an optical barcode of 1, while some other transcripts in this particular round, there is no fluorophore attached, then it will be fluorescent off, and then it will be decoded as 0. And by doing this in multiple rounds, then it started to build up this binary barcode. And with this, then it really helps to resolve the identity as well as the quantity of different RNA species. So to really ensure that we can detect each transcript very accurately, MERFISH actually has further sort of taken into consideration of error correction using this error robust barcoding scheme as illustrated on this slide. So basically, each barcode sequence, this sequence of zeros and ones, are designed to have enough space in between them so that incorrectly assigning a barcode is very unlikely. Even if there is a readout error as illustrated here, the system can actually assign the nearest correct barcode and actually detect and correct the mistakes. So thereby, that's why it makes MERFISH or MERSCOPE measurement so accurate and consistent. So using this barcoding system, MERFISH really gives us the flexibility to create the custom gene panel. And this custom gene panel can go from hundreds to even thousands. And this spatially resolved RNA profiling data now started to really give a physical picture from the cell or tissue of interest, as pictured here. Now these colorful dots indicates different RNA species in the cell, while the gray lines indicates the boundaries of the cells. You can really start to zoom out at the whole tissue level to get a sense of how genes are expressed spatially, or zoom in into individual regions or any region across the tissue. And then to understand the gene expression profile within single cells or even at subcellular resolution to get a sense of how different RNAs are distributed within individual cells. So MERFISH was initially published from Xiaowei Zhuang's group in Science back in 2015. So since the initial publication, MERFISH has been validated by a long list of publications as listed here, and has already brought many transformative discoveries. And actually, spatially resolved transcriptomics was selected as method of the year by Nature Methods in 2020. So MERFISH was also highlighted, really highlighting the excitement it has already generated in the field. So to really make this powerful technology more broadly available in the research community, that's why Vizgen started to step in to commercialize MERFISH through our platform called MERSCOPE. It really facilitated the entire MERFISH measurement with the complete reagent, instrumentation hardware, and software solution. So upstream-wise, we have ready-to-use MERFISH gene panel and sample prep reagents to stain and process the samples. And then the processed samples will be able to load it onto the MERSCOPE instrument for automatic imaging, and then downstream, once the data is decoded, then it can be plugged into these very interactive visualization tools to allow data analysis and visualization. So in the next few slides, I'm going to really walk you through the MERSCOPE workflow first, starting from the gene panel design. As you can imagine, as a targeted RNA imaging platform, so gene panel first need to be specified and designed first. And we have developed a web page-based portal, as shown on this slide, to really help everyone to construct the custom gene panels. And then Vizgen will be able to provide ready-to-use gene panel reagents for everyone. And then from here, then it will start to create a code book and assign the relevant barcodes to the gene panel. And then the sample will be prepared on the MERSCOPE slide and undergo these few steps of workflow. First is the fixation and permeabilization of the sample. And then some samples may need a cell boundary staining to really enable single cell analysis. This is a protein-based immunofluorescence staining to basically stain the plasma membrane of the cells. And then downstream wise, then the algorithm will be able to really help segment the cells and enable downstream single cell analysis. And then after the antibody staining, then comes with the hybridization with the DNA oligo pool. And then we have additional 2 steps for removing the autofluorescence background. And that really makes the signal to be easily picked up by the decoding algorithm. So overall, the procedure for preparing and processing MERSCOPE samples is very similar to a standard in situ hybridization protocol with hands-on time actually only about 1 to 2 hours each in total. And we believe that any researcher will be able to adopt it very easily. So with the samples processed, then it will be assembled into a flow cell as pictured here. Basically, it will come together with the MERSCOPE, and then it will be put together onto the imageable region, basically onto the high NA objective. And then for each gene panel, it will have its own reagent pack that again will be loaded to the MERSCOPE as well. And then you will be able to sort of really specify the region of interest to image. And for MERSCOPes, here are some of the key features or specs of the instrument. Basically, it has a very large imaging area of up to 1 square centimeters, and it uses very high NA objective. So it has imaging resolution of 100 nanometers. So then on this slide, it shows sort of the instrument software. Once the sample is loaded, you can use this software to set up the run. It usually takes about 20 to 30 minutes to set up such a run, and once set up, the instrument will take care of the downstream acquisition automatically. And then comes out of it are actually 5 different data outputs. So the first one is the list of all detected transcripts, which contains the genes, the counts, and XYZ coordinates. Then the second one will be the mosaic images, which contains the DAPI or the poly-A or the total mRNA stain of the sample, as well as some other protein staining. For example, if you included the cell boundary staining or some specific protein marker imaging, then it will be included in this high resolution TIFF file and show up as a mosaic image. Then the third one, the transcripts per cell matrix, is very similar to a single cell RNA-seq data output, which contains the gene and cell matrix, the gene counts per cell and such. And finally, since this is a spatially resolved measurement, we do provide cell metadata that contains the XYZ coordinates of each cell, as well as the volume of the cell. And then the polygon shape of each cell is also included in the last format, the cell boundaries, which is a HDF file. So you can use, for example, software packages such as illustrated here, ScanPile Seurat for single cell analysis or downstream visualization. Alternatively, within Vizgen, we have also developed MERSCOPE Visualizer, which is shown here. This is the interface to really help facilitate the data analysis and visualization. And this is directly comes with the MERSCOPE as well. So as a gene expression profiling tool, as you can imagine, MERSCOPE at its basic form just reports the location, quantity of RNA species, and the cell boundary information. And with so much information, it really started to open up many application areas. And we envision that, as illustrated on this slide here, that it can be applied in many research fields. And in the next few slides, I'm going to walk you through a couple of showcase examples to really illustrate how we could use MERFISH to gather new biological insights. So the first showcase example illustrates how MERFISH can be used to profile gene expression in situ across very large tissue areas. And as you probably know, that to understand the tissue function, sometimes it's very critical to understand how different genes are expressed at different anatomic sites, how the expression changes during disease onset or progression, or even after drug response. So in this particular example, we used MERSCOPE to profile a panel of around 500 genes to really map out the cell types as well as cell receptors in the mouse brain. And it includes 50 canonical marker genes and 433 GPCRs and RTK. So it's a mouse receptor map project. So on this slide, on the left, what you're seeing is 8 RNA subset out of this 483 gene measurement in a 9 by 7 millimeter image. And hopefully just looking at a very small subset of genes, you can already start to see the very structure, the very intricate structure in the mouse brain here. And since this is a very high resolution image measurement, you can zoom in into any regions of the mouse brain and actually started to actually get a sense of how different genes are expressed in single cell level. For example, shown here now, what you are looking at is actually the expression of all genes, and hopefully you can start to appreciate some of the difference of gene expression. For example, this cell I'm pointing here has very different expression profile as compared to some adjacent cells. So notably, MERFISH is a very quantitative and highly reproducible measurement. We often benchmark the performance of MERSCOPE versus some of the other technologies. Shown here is the correlation with bulk RNA sequencing. And hopefully you can see that actually it correlates very well with the correlation coefficient of 0.88. And importantly, the FPKN value for the measured genes spans quite a few orders of magnitude here. We have the ability to measure genes that are less than 1 copy per cell, as well as genes that have FPKN of approaching 1,000. And in between biological replicates, this is a highly reproducible measurement. And as you can see by this correlation plot here, this has a correlation coefficient of 0.99 here. So to really illustrate some of the key features of MERSCOPE versus some other spatial technologies here, we're trying to compare the performance of MERSCOPE versus the oligoarray-based approach in matched brain slice. So again, hopefully by looking at this, you can sort of start to see how it compares the data quality for MERSCOPE. And very notably, MERFISH actually, if you look at the correlation plot here, well, versus the array-based approach, MERFISH consistently detects 70 times more transcripts per gene as compared to array-based platform. You may start to wonder what this 70 times more transcripts really mean in real biology. So I sort of really come here to put some context and some concrete numbers first. So in academic publication, MERFISH has been demonstrated has a detection efficiency of around 95%. So for a gene that expresses around 100 copies per cell, that means that there will be 95 copies detected by MERSCOPE. However, if you have a drop of more than 70-fold of detection efficiency, then a gene that expresses 100 copies, you may only have the ability to detect 1 to 2 copies. And that actually manifests here in one of the moderately expressed gene, OPRD1, which is an opium receptor delta 1 gene. As you can see here in the top panel here, using the array-based approach, there is only a few grid spots light up for OPRD1, while in MERFISH, you started to see that this gene actually has a very well-defined structure in different regions of the brain. And furthermore, in addition to the high resolution, the high detection efficiency, MERSCOPE also features true single-cell resolution. You can keep zooming into any region of interest and really started to have a zoomed-in view of how different genes are expressed in single cells. For example, for OPRD1, again, looking at the zoomed-in image on the right, you will be able to see in this particular 200 by 200 micron image where OPRD1 is expressed in individual cells. For example, you can see that a couple of cells are light up in green here. Well, as compared to the oligoarray-based approach, this single-cell resolution is completely missing because a grid spot size is usually around 55 micron size, and then in between, all the information is actually missing. So in short summary, it really offers true single-cell resolution, and with that, you can really start to perform true single-cell analysis. And as shown on this slide, by clustering the cell types based on the gene expression profile, the MERSCOPE enables us to do, for example, a UMAP clustering shown on the left. Now this is showing the dozens of different cell types identified by MERSCOPE. And on the right, since this is a spatial measurement, so all the X, Y coordinates of identified cells can be registered back in situ. So now all these different colorful dots indicate where individual cells are located in the brain regions now. So the second example illustrates, actually builds upon the cell clustering capability of MERSCOPE and actually goes a little bit beyond, and it's sort of illustrate how we can use MERSCOPE to not only map cell types, but also characterize cell function and state in mouse brain. And this is a study published in Science back in 2018. And in this particular example, MERFISH was used to profile a panel of 155 genes in the mouse hypothalamus region, pictured in this blue square here. And by profiling almost 500,000 cells. So it really features very high cell throughput. The authors were able to identify more than 70 different cell clusters in this particular brain region. Again, all the major cell clusters are highlighted in different colors here on this T-SNP plot. And again, the authors were able to compare the single cell clusters identified by MERFISH versus single cell RNA-seq, and it correlates very well with single cell RNA-seq. And in some instances, MERFISH was even able to resolve cell clusters not identifiable by single cell RNA-seq. For example, here, there were 2 clusters that were grouped into a single one by single cell RNA-seq, but were resolved into 2 different clusters that are actually spatially, they are at different locations in the hypothalamus region. And this is really made possible by 2 key features of the platform. The first one, as I mentioned, is the high cell throughput. By sampling more cells, it really allows rare cell types to be captured. And secondly, since the platform has very high detection efficiency, it really allows many lowly expressed genes that are important for specifying cell types. And in this particular study is the neuropeptides to be very accurately profiled by MERFISH. And again, since the location of all cells are recorded, the authors were able to reveal the cell organization within native tissue context. I'm showing here the colored dots indicate different cell types from anterior of the mouse brain to the posterior of the hypothalamus region. And hopefully you can see the very heterogeneous distribution of different cell types across this few hundred microns section of the hypothalamus region. And furthermore, by including activity marker genes, and in this particular example, a C4 gene that reports neuronal activity, MERFISH was able to identify which specific neuronal types are selectively activated during individual social behaviors, for example, parenting behaviors, mating behaviors, and the authors were able to really pinpoint where those neuronal clusters are once they're activated by these social behaviors. So hopefully this example really illustrates how we can use cell atlasing capability of MERFISH platform to really not only define and catalog cell types, but also characterize the cell function. So the next example that I'm going to walk through is a more clinically relevant sample. And here, MERSCOPE was used to profile a panel of 417 genes in human colon cancer. And what you are seeing here on the left is the spatial distribution of 10 RNA subsets out of these 417 genes. And just because it's difficult to show hundreds of color images, so we picked only a couple of genes highlighted here. And with just a few genes here, I hope you can already start to see the very rich spatial information for different genes within this tumor slice. For example, we can see a group of SOX9 positive tumor cells encircling the SPP1 stroma cells. And below these are a group of immune cells marked by the RGHA1 gene. And in this measurement, since the cell boundary stain was performed, so we were able to really segment the cells and identify individual cells, and then that allows us to really zoom in into individual cells and interrogate the gene expression profile among individual cells, as shown by this heat plot here. And with that, again, we were able to really perform single cell clustering and identify all the major immune cell types within the tumor slice. Shown below on this picture, all the major immune cell types identified, including CD8, CD4, NK, dendritic cells and such. And hopefully by looking at the animation, you can see the different spatial location of these different immune cell types within the tumor. And in a very similar application area in this 2021 publication in Cancer Cell, MERFISH here was used to explore the crosstalk between glioblastoma and immune cells within the tumor microenvironment. In this study, the authors will actually perform single-cell RNA-seq, and they were able to identify 4 major malignant cellular states in glioblastoma. And then they used MERFISH to profile a panel of 135 genes. Very importantly, they were able to recapitulate all the major cell types bound by single-cell RNA-seq, and very specifically shown here on this heat map plot here. These are some of the major cell classes identified either by single-cell RNA-seq or MERFISH. the authors were able to find that there's a very high correlation between MERFISH counts and then the corresponding bulk RNA sequencing data. And since the spatial information is recorded, MERFISH now really started to enable the authors to investigate the spatial localization of different cell types. Very interestingly, they were able to find enrichment of macrophages adjacent to MES, like glioblastoma cells in situ. So the next example here, pictured here, MERFISH was used to study kidney injury, and this is studied done with Dr. Ben Hammersnap at Washington University at St. Louis. And here specifically, MERSCOPE was used to profile a panel of 243 genes in mouse kidney, and adjacent kidney size was profiled by array-based platform. And since this study hasn't really been published yet, so I'm only highlighting one gene, SLC12A1 here. So this gene is a kidney-specific sodium-potassium chloride co-transporter and is enriched in, for example, in macular denser region. And hopefully, by comparing the two pictures, one on the left is the array-based platform, on the right is the MERSCOPE, you can see the striking difference of the information that you can get from a MERSCOPE. Basically, on the left, you don't really see too much detailed information for the expression of SLC A1, while basically on the MERSCOPE, this gene are lighting up the very intricate structure in kidney. So in other words, the high detection efficiency of MERSCOPE as well as the high resolution provided by MERSCOPE really enables spatial profiling of gene inaccessible by some other commercial platforms. And of course, there are many other new insights that comes out of this study. It's just for now, we cannot really disclose any details yet. So in the last showcase example here, MERFISH was used to investigate the tissue architecture in liver. And this is a study published by a group, Stephen Wang's group at Yale University. And here the authors were interested in understanding the hematopoietic stem cell niche and the loss of function of TET2 to really understand some of the hematopoietic diseases. And in this particular study, they used MERFISH to profile a panel of 140 genes, and they imaged thousands of tightly packed cells. And as shown by these clustering results on the right, they were able to identify 8 major cell types and determined the spatial location in the liver. So very importantly, several cell subtypes, for example, the AECs and ACCs were identified by MERFISH, but actually were missed by single-cell RNA-seq. And the authors compared these subclusters of cells in wild type versus the knockout mouse, and they were able to discover actually a switch of Wnt and notch pathway genes in these the subtypes of cells in the knockout mouse. This is something that the authors were not able to do by single-cell RNA-seq. And again, it really highlights how the high detection efficiency and accuracy of MERFISH can really enable new biological discoveries that is not possible by traditional technologies. So I hope all these examples that I have just walked through have given you a very good impression of what MERFISH is capable of. And within Vizgen, we keep pushing the limit of our technology by further expanding its application areas. We have now proven MERFISH in more than 20 different tissue types. And on this slide, I'm only highlighting a couple of representative tissue types here. For example, the human ovary sample using a panel of around 300 genes, the human spinal cord, you can see the horn structure here, the mouse intestine, the human kidney, the mouse lung sample here. Again, on each, only a subset of genes are highlighted because it's very difficult for us to show hundreds of color imaging here. And among all these pictures, my personal favorite is actually this mouse small intestine sample. Is more like a very, very elegant seahorse type structure. Actually, this group of cells are immune cells that are enriched here, while you can also see the intestine NIAP structure here for this picture. And finally, we really aim to make MERFISH compatible with any sample type. And so far, in-house, we have validated MERFISH in cultured cells. This includes adhesion cells or even liquid biopsy samples. And then the tissue blocks includes the fresh frozen and fixed frozen tissue blocks. So we really hope that this versatile tool could be added to any researcher's toolbox to help accelerate the research. So to just recap what I have just shared, here is a really quick summary of what I went through today. So some of the key advantages and specs of MERSCOPE. First is the high multiplexing power. For a single measurement, it covers hundreds to thousands of genes in a single sample. And secondly, it has really high spatial resolution. And we can localize RNA transcripts with subcellular resolution. And this very high spatial resolution is preserved across the entire tissue. Third, MERSCOPE has the highest sensitivity for identifying RNAs, and it really enables us to detect lowly expressed but functionally very important genes. And fourth, it features very high cell throughput. We can profile a square centimeter sized tissue with hundreds of thousands, even sometimes millions of cells in a single sample. And no follow-up sequencing is needed for MERFISH or MERSCOPE run. You get all the information through a single run. There is no additional sequencing cost. And finally, this is a very flexible platform, and you can essentially run on any sample or tissue types. So this is, again, just a recap of what I just said. If you're interested, I can later post on this slide. And in very short summary, I hope this presentation gives you a good overview of our platform. And we're really excited about the potential of this technology and would love to hear how we could potentially help with your research. And with that, I wanted to say thank you again for inviting me for this seminar. And if you have any questions, please let me know. Or here, there is also some contact info that you can reach out in the future as well. Thank you.
Thank you, Jiang. Yeah. So I see a few typed in, so I'm going to read the Q&A or chat. So among the participating members in the audience, please send your questions either by typing in or use the raise hand tool. I will be able to call you out and unmute you. So Jiang, I don't know if you can see it, but I can.
Sure, yeah, I can see the Q&A. There is one question about the number of genes. So would you mind telling more about the best practices for selection features for profiling? So in terms of the MERSCOPE, the reagent, the first generation reagents that we released, we have three kind of gene panel. It features 140 genes, 300 genes, and 500 genes. And the cost will vary, depends on the number of genes that you select. So again, this is something that we envision that we will further expand the gene panel. So since this is a complete, our default offering is a custom gene panel, so it really depends on your biological question, how many genes are needed. So oftentimes through our current clients and lab service program, we found that 140, 300, or 500 usually meets the, for example, for cell atlasing purposes, is already quite sufficient. But of course, we envision that for more discovery-based approach, higher gene number may be preferred, so that 500 gene panel one may be sort of a better choice if it's more for discovery. And if you're interested in hearing more about the pricing, I think you can sort of reach out to our marketing team. They have more pricing related info. So the second question is, what are the options available for users who want to perform MERSCOPE on the sample? Do they need to buy instrument or submit samples to us directly? So we do have two options available. The first option is the instrument and reagent model, where actually end of August, MERSCOPE is already released to the public, so it's commercially available now for purchase. And we actually already have some of our customer base already having MERSCOPE in-house now. That gives you the flexibility to basically run MERFISH on your own. At the same time, we do have a lab service model where our clients basically submit the sample, and then we will help to design the gene panel, process the samples, and deliver the data back. So this is a second option that can go for. And again, if you're interested in sort of running service with us, you can also reach out to us through the email contact here.
So Jiang, before you moving on to the written questions, I unmuted Eugene. So Eugene, you raised your hand. Go ahead with your question.
Thank you. It's beautiful talk. The questions from me, if we are interested and to buy the equipment, and can you share a little bit more information? What is the competitor for you people and why we should go for you company? I know you did a great and very impressive talk here.
Yeah. So I think a couple of key differentiating factors that I can further highlight, because there are a couple of spatial technologies that are currently commercially available. For example, the Vizgen or the Genomics. I think for now, MERSCOPE is actually the only sort of true single cell platform, and it has the highest detection efficiency. I highlight the currently commercially available platform. These are probably the two key features. The first, the resolution-wise, as I highlighted in some of the showcase examples, you can see that the array-based platform really has great spot size of tens of cells and 55 micron size. So that is one of the big difference, and there is no way to really tell where individual cells are. While MERSCOPE has imaging resolution of 100 nanometers, so that's one key difference. The second one is the detection efficiency, basically how many genes that you can detect out of the tissue that you're running on this scope. And since we compared actually in two cases with matched sample, actually even adjacent tissue size, we really see the very different detection efficiency for our platform versus the others. But again, these are some of the sort of technical aspects of our platform that I can highlight. But of course, if you're interested in hearing more about some of the other features, happy to chat more as well.
Jiang, you can go back to the Q&A. If you can do 1 minute per question, well,
sure, yeah. So for human skin, we haven't actually started working on human skin yet, but our experience indicates that as long as the sample's RNA quality is high, we usually don't have too many issues. So the next question. So for imaging illumination and magnification, so we are actually doing 60x for high resolution imaging, and we are doing 5. MERSCOPE has 5 laser channel. So for Murphy specific imaging, we're doing 3-color imaging, while the Polity and DAPI are used for some other purposes. So the typical time to cover 1 square centimeter of tissue, so this really depends on the gene number and the area size. For example, for 500 gene panel for up to square centimeter, the experiments usually will be finished within around 24 hours. However, if you do 140 gene, this will be finished around 18 hours or so. No, for up to a square centimeter. So next, the multiplex scale up. Can you effectively multiplex or scale up the sample prep? Yes. So the sample prep, yes. So the MERSCOPE instrument takes 1 sample at a time. However, the samples can be prepared in batches. For example, batches of 10, and then it can be stored for quite long term. So you can once finish one and note the next one. So usually there are multiple stopping points actually upstream for sort of fixation step. There's certain steps that the sample can be stored. After the sample is ready for imaging, it can be stored in our last solution for up to a week or so, or even longer. So next, is it possible to combine mRNA detection with protein detection using this platform in the future? The answer is yes. We actually are routinely doing this now. And we are hoping to release a low-plex protein detection product offering, hopefully in the very near future, because for now we have already routinely do it through our lab service program. Next, so more information on frozen tissue accepted is OCT tissue. So yes, so for the tissue types, currently the platform is compatible with fresh frozen, snap frozen, or fixed frozen. And the default is actually the sample need to be embedded in OCT for cross-stat sectioning. So the tissue will be sliced into 10 micron thick. And internally, we're working on FFPE tissue, and we have so far got very promising data indicating that even for FFPE sample, if the RNA quality, so we have different metrics for evaluating RNA quality. The data is actually comparable with the fresh frozen sample we have now tested on multiple, for example, mouse brain, liver, lung, and all are showing very good quality even in peripheral embedded samples. But for now, we haven't really released the FFPE product to the public yet. So how is the segmentation performed and how accurate it is? So for the segmentation, there are usually two approaches for central nerve system. For example, the example that I was showing here, the brain and the spinal cord, these are central nerve systems. We usually use the DAPI and total mRNA, use these two signal to segment the cells because the cells are very separate from each other in central nerve system samples. However, as you can imagine, for very dense tissues, as illustrated here, for kidney, small intestine, all that, we are using cell boundary staining to first really define the cell boundary, and then later the algorithm will be able to pick up the best cell boundary staining for analysis. And again, here, there are quite a few options, and the users usually have the option to evaluate what is the best before they decide which one to go. So the last question is the lab service. What is the turnaround time? So currently our lab service program is quite oversubscribed, and we have quite a long waiting list at the moment. It usually divides into two phases. The first phase is to, if it's a tissue type we have not really worked with, we oftentimes need to do some protocol optimization to do other protein staining optimization or gene panel evaluation. That usually takes several weeks to a month or so. And then once the protocol is defined, then the next is the sample processing, and usually again just 1 month or so, 1 to 2 months-ish, I'd say, depending on the sample type, sample volume, how many samples will be processed.
John, you have some questions in the chat as well.
Oh yeah, sorry, yeah.
I had a very quick question. Is it okay, June? Can I?
Sure, Vanessa, go ahead.
Yeah, very nice talk, Jiang, actually. And, thanks for taking time to give this presentation. I was wondering about how many rounds of hybridization you need for a typical couple of 100 genes, and what's the sample prep time, along with the imaging. That was a question I was curious to, but somebody asked that. Thanks.
Yeah, so for example, for 140 genes, it only needs 6 rounds of imaging. For 300 genes, it needs 1 additional round, 7 rounds. For 500 genes, it's a total of 9 rounds. So it scales very favorably because of the barcoding scheme we have.
Great.
And the setup time, so the time for imaging usually, for example, depends on the tissue size. For square centimeter, it usually takes about 2 hours or so, or a bit longer, to finish scanning the entire area. And then it has the reset time of around 1 hour or so, and then it goes next round of imaging.
Thank you.
So in the chat, Darwin, hello. Yeah, so what degree the null calling rates for null expressing genes using the array-based detection technology? So again, this is tied to the detection efficiency for the array-based technology and the MERFISH. I think one of the key differences that really makes the detection efficiency different is the different chemistry used, because here MERFISH is using direct hybridization based, so there is really no other sort of indirect process to detect the genes of interest, while the array-based one, first, the RNAs has to be released from the tissue and hybridized to the array first. And that step is not going to be sort of super effective, depends on how the enzyme and some of those digestion steps are capable of releasing the RNA to be bound to the oligo array. And downstream wise, then there's sequencing-based amplification, and again, that again will cause some of those drop of detection efficiency. So there are, since the chemistry between these two technologies are very different, then that's sort of making why the detection efficiency differs so much. So the next question is, does the platform know the construction of 3D structure? So for now, actually, the platform itself does do Z-scan, but we only do sort of 10 micron thick tissue slice. So we do a couple of these slices, and that does know the 3D reconstruction. But in order to construct hundreds of micron or even thicker ones, that requires serial sectioning and sort of remap everything back. So next, so finding cell subtypes, did we try to cluster the single cell RNA-seq or using the same select genes down? Yes, so actually a couple of already published data using MERFISH versus single cell RNA-seq, very similar clustering or even sub-clustering were performed to really benchmark the performance of MERFISH's ability to identify cell types. Sometimes in order to define the major cell clusters, it's just sort of the UMAP or T-SNE clustering is not really done in such a way that you resolve too many. And of course, you can always sort of group, say, sub-clusters of cells and then keep doing some sub-clustering. For example, this 2018 paper in Science, the authors will actually pick out those in excitatory neurons, inhibitory neurons, and then they were able to do sub-clusterings of the cells, and then that actually allows many new cell types to be identified. So indeed, sorry, and then the same selected genes were also used for clustering. And in this particular study, the authors were actually able to find that in order to find these 75 different cell types, there is really no need to use 155 genes. Around 75 genes is sufficient to actually find and identify and recapitulate all the cell types found by single cell RNA-seq.
Yeah, John, I think we reached.
Yeah, I think that's all the questions.
I was going to say it's 2 o'clock now, but this was a fantastic presentation. It seemed to generate a lot of interest, and so thank you so much for your time. Did we get everybody's questions, or are there outstanding ones? And
I think so, Vanessa. I think we reached. The last question was reposted asking for how many genes. I think right now it's a few hundred. You know, there are panels of 500.
Yeah, we're hoping to scale this up sometime in the near future. So definitely higher genes to put this on our roadmap. It's just our first generation of products. For now, we found 500 is often sufficient for many biological questions.
Right. So given the time, I'm about to close it. Jiang, thank you again for meeting with us. Thank you, Vanessa, for organizing it. And I'm sure there will be other questions. Please reach out to us and we'll forward to you. And Jiang, also thanks to your colleagues in Vizgen. Hope to see you soon.
Thank you.
Definitely. Thank you, everybody. If you have any questions, don't hesitate to reach out to us.